And our next speaker is Betzig at Berkeley and also partly at Janelia.
Okay, it's probably apropos that I, as a microscopist, am giving a talk about AI today, because after all, it is April Fool's Day. But at least I can rely on a lot of help from my friends, and hopefully I don't sound like too much of an idiot in the end. So much of what we know at the subcellular level comes from these three pillars of modern biology. The problem with them is that they're all reductionist approaches that are about taking the cell apart and looking at its pieces. And I contend that you cannot understand any dynamical system from taking it apart because you have all these unknown missing parts and so forth. It would be like trying to understand a car engine from a random pile of engine parts. And, oops, let me turn off sound here if I can. Turn it down. Okay. So as a result, the human mind is forced to try to figure out or hypothesize what are the missing pieces in any process, and then to hypothesize what are the dynamical relationships between those pieces. And so as a result, for example, with transcription, by 20 years of molecular biology, a model formed for how different transcription factors come together and over the course of minutes to hours to eventually recruit a polymerase, which then creates the RNA. However, 1 second of live imaging by single molecule tracking demonstrated that none of these transcription factors bind to the DNA more than for a few seconds at a time. So what finally emerged from this was a stochastic model of how transcription occurs, very different from what was hypothesized from what we knew from biochemistry. So I contend that there's a lot you can learn from biology by just damn looking at it, okay? And so that's basically why I'm a microscopist. So in particular, there are two conclusions. First, for any subcellular process, a holistic approach is the only way we can ensure that all the players are present, the seen and unseen ones. Second, animation is really the hallmark of life, and only optical microscopy has the gentleness, the resolution, the speed, the sensitivity, and the specificity to be able to interrogate it at many different levels. And so the overall point is, how can you understand life if you don't look at it live? Now, traditionally, microscopy has been applied to biology in the form of static 2D images or 2D movies because that's what our eyes and brains were evolved to handle. However, cells exist in 4 dimensions and can really only be properly understood in those terms. About 15 years ago, my lab developed a lattice light sheet tool that allows us to non-invasively study small cells or small organisms at high resolution in 4D for long periods of time. However, cells in culture are very different from cells in tissue, and true physiological relevance requires studying cells in their parent organisms where all the environmental cues that drive gene expression are present to make sure that we're looking at the native phenotypes. The problem of doing that is that in multicellular organisms, the light is scattered and aberrated by the tissue itself. And so around 10 years ago, we borrowed from astronomers a technology of adaptive optics to allow our microscopes to correct. So we go from an aberrated image to a corrected image here inside of the brain of a zebrafish to look at the neuron structure at the original resolution we would have if we had pulled those neurons out of the system. So we combined our adaptive optical techniques with our lattice light sheet techniques, and then we had a tool that would allow us to study subcellular biology and dynamics inside of living tissue, inside of the whole developing organism in different types of systems. And this technology covers an enormous range of space and time, 7 orders of magnitude in time, 10 orders of magnitude in volume. So you get lots and lots of diverse data. Now, because you have so much scale and so much speed of the microscope, it means we can collect data with one microscope at about 4 terabytes per hour. And so it was necessary for us to develop a computational pipeline that would allow us to be able to deconvolve, stitch, compress, and store all of that data in real time at that 4 terabytes per second rate. The problem then is where we're stuck, is we can take massive amounts of high quality data, but the problem is what do we do with it? How do we analyze it? So in a couple examples, we spent about a year and about 3 million CPU hours each to try to extract some quantitative insights from 2 of these data sets. But it's a tiny fraction of all of the data and a tiny fraction of all the questions you could pose. And furthermore, we were influenced by this problem of data by 10 years of trying to get our tools in the hands of other people through either open access, like our AIC here, open sourcing our microscopes and the designs, or commercializations of our technologies. All of these methods have largely failed to have much impact. And the reason is because people can't handle the data that these microscopes produce. We have a collaborator we were working with 10 years ago who just submitted a paper on data that they were analyzing for the last 10 years. This is the problem we're at, and this is why we need AI, okay? So we want to have some kind of model to understand subcellular biology. It's a 5D problem, X, Y, Z, T color, okay? So it's incredibly far along the curve of where AI models are today, but it's the only way to start to deal with the complexity that exists. And coming as not a biologist, one of the things that still gets to me every day that I use my microscopes is how incredibly complex life is. And when I hear talks about transcriptomics or so forth, it is such a reduction of such a more massively complex thing. And we have a lot of work to do, is bottom line for it, in order to understand all of this. Now, the one format which has been successful, one thing that's common about AI models that have succeeded in different tasks like large language models or AlphaFold is that they require vast amounts of well-curated data, okay? Well, that's the only thing that I contend that I'm really good at is I can take lots of really good data in quantity, okay? So it requires, if you're going to use a transformer architecture that is a good one to use potentially because it understands the spatial temporal relationships that are in the data. This is a spatial temporal problem we're talking about. It's suited to large tasks, machine level tasks, but it requires vast amounts of pre-training data. We can take that. Requires substantial amounts of annotated data, which is harder because you have to find out a way to do the annotations. But it's also extensible then to multimodal foundation models where we could take all sorts of orthogonal type of data as well and put it into the model. And so if we had such a model, again, having worked 15 years now with probably 150 different biological groups on different problems, everybody has a different question, everybody has a favorite protein, everybody has a favorite system. So if we had such a model, you're going to have an incredible diverse of questions people are going to ask from the molecular level to the tissue level. And so you want to be able to build a model that can generalize across all of those scales. Now, if you look at this, the 2 rightmost columns involve things happening between single cells or cells in combination. Things on the right are what's happening within individual cells, but they're still defined as cellular compartments. And so of all the tasks that you'd want to do, the first one you'd want to do is segmentation, okay? So we've heard lots of talks about segmentation. It's coming along, but what we need is true 4D segmentation in order to do what we want to do. But if we had that, then this is the gateway task to be able to understand the complexity of the data, because after it's processed, if you render it, it's going to look like that thing at the left. And if you really want to understand it, we got to tease that apart. So we started to benchmark segmentation tools like CellPose and PlantSeg and MicroSem for fairly simple data inside of a zebrafish embryo It's about, on a per time point basis, about 70% accurate. But if you then try to follow those cells over time, you get lost very quickly, as you can see at the right. So there's a lot of work still to be done. We've also, to try to benchmark it against the annotation tools that have been developed here for FlyEM, in order to try to see what time scales would be involved for that type of annotation. Well, here's a little tissue section that we took over 10 hours. It's less than half a terabyte. We asked them to segment 3 cells over a few time points. Every time point, every cell averaged about 90 minutes of manual annotation to do. If you try to scale that up to the whole tissue, just this little teeny chunk of tissue that we took in 10 hours, it would take 17 man-years in order to do that. Not scalable. Okay. So our approach is first to develop, let the organism itself to help in the segmentation process, to create annotations. And you can do that with a rainbow-like technique we call Skittles. We've developed with 5 different fluorophores that are stochastically expressed in different cells. So those are real images then of the colors that we get. And we will use that as with a lot of work on finagling exactly how to create labels out of that data to create the annotated data for the segmentation task. Once we have the segmented data, though, then one can ask, like I said, an incredible array of different types of questions. We're starting with zebrafish, but everybody's going to have their own ideas about what they want to do with different types of organisms. And to the extent the microscopes are capable, we'd like to be able to follow along with that. In order to get our feet with understanding what would be necessary to create a 4D transformer, vision transformer, we started with the problem of developing a 3D vision transformer, specifically to do aberration measurement on zebrafish based on endogenous expression of clathrin-coated pits in the fish as fiducials in order to do that. We were able from that then to determine the scaling laws for data collection, for training time, for compute, for inference time, and get a robust model that could, given that you have clathrin-coated pits, you'd be able to do accurate estimates of the aberration. And we learned a lot about exactly what's the difference between how much time it takes for final training of the model versus the much larger amount of time you try to do training of different model architectures, finding the right model size and all that, which greatly explodes the amount of compute that you have to do. But the final message of those things, if we try to extrapolate that to 4D, was that we're within the ballpark now with the state-of-the-art of compute to be able to do pre-training for these types of tasks. In 4D, we start off at where sort of the largest vision transformer models left off and then go up from there. So with sufficient development of the technology on the hardware side, we think we can march up this curve. We think we're hitting the right complex problem at the right time in the technological development curve. From our work with that 3D model, plus also what has been done with VIT and SAM and other things, we can kind of guesstimate what are the needs in terms of data collection, in terms of compute in order to do first the pre-training, and then the one of the tasks, segmentation, and then other tasks would be similar in terms of the amount of compute. So for pre-training, we need less than a petabyte here. On our scope, that takes, maybe 150 days per scope. So we've been trying to develop very performative microscopes to take data at the scale that we need to do it. The first one is based on one we've had in hand for a while, our Mosaic microscope, which does adaptive optic lattice light sheet. It works quite well. We get high quality data. That was a slab, a MIP slab. This is a tinier MIP slab, so you can see internally. This is in the spine of the fish. You can see it actually swelling as it's growing over 20 hours, from 24 hours to 44 hours. 4.2 minutes to get the whole volume. Resolution about 220 by 220 by about 400 nanometers over that whole volume with pretty good time resolution, 20-hour time lapse. This was taken last week, so it hasn't been prettied up, the data, that much. But I'm trying to express that people have talked about needing large data. We've got large data. We can get tremendous amounts of quality large data. However, this particular microscope has a limitation that that because of the large chamber and so forth, it's not amenable to doing perturbation studies. And it's good for doing a long time lapse, but again, for your models, you want diversity. So that means you don't want to spend a long time on one fish, you want to spend littler time on many fish, and it's just kind of a pain in the neck to go in and out. So we're developing now here at Janelia an inverted version of this microscope with an open top for perturbation, where we can lay multiple embryos across there and then kind of in a more industrial sense be able to start to image that. It would have a little bit more performative resolution, longer working distance to go deeper in the fish, and multiple cameras to speed up the process there. And also then at Berkeley, we applied a swept oblique light sheet microscope I developed for my startup for single molecule tracking to looking at the fish with it. And even without AO, this is looking in the eye of the developing fish, it was pretty performative. And so the nice thing about this microscope is it's in a format which actually is on a standard inverted frame, can take multi-well plates, so we can line up many embryos, do many perturbations, and go right across and study them all one after the other. And so we're finishing the design of this microscope now and should have it running by the end of the summer. So we should have 4 microscopes, each capable of taking on the order of 5 to 10 terabytes of data every day. So comparing microscopes to point scanning microscopes, the point scanning microscopes, area scan, confocal, 2-photon, aren't even in the conversation in terms of the data rates necessary to generate large data. They also, of course, bleaching rates are enormous, so you wouldn't be able to get very big volumes. But with the lattice light sheet technology or the ISOR technology, we can get data as good as any cell you've ever looked at on cover slip and tissue. So the ultimate goal then is to develop the models in this fashion, where people have a vast range of questions they can ask. Either if those questions are based on data by which the model has already been trained, then that can be asked through an LLM, and the vision model will then be queried by the LLM and then produce human interpretable results for people. If they have something new, then we can do retrieval augmented generation with additional data to then get new questions answered by the model after that. And so what are the kind of questions? I mean, again, I am not a biologist, but as a physicist and as an engineer, it has always worried me. To what extent are the morphologies we see on a cover slip recapitulated in multicellular organisms? How do they differ in different cell types, different phases of the cell cycle, different developmental stages, different organs? What's the one of the things discussed yesterday in the discussion was making AI models that can either predict or understand. To me, this is, I think in terms of that single molecules doing the transcription. I view the whole cell as properties of emergent stuff off of stochasticity. And everything that happens in biology follows a distribution. It's not deterministic. It follows, you have to understand the distributions. There's not one outcome, there's many outcomes. We need to understand those distributions. How are these distributions affected by perturbation? How do different events at the subcellular level manifest themselves at higher levels, from subcellular to individual or collective behavior of cells in development? What's the spatiotemporal relationship between the transcriptome and proteome in the phenotypes observed at all these different levels? Can our observations allow us to ultimately predict behaviors or responses to perturbation? And lastly, what spatial-temporal relationships might the model itself discover in its latent representations that would be unknown or invisible originally to the human eye? So I think there's a lot of wins that can come out of developing this type of thing to answer some very long-standing questions, and that's what we're trying to do right now. Thank you.
So do you think there will be a time that there will be enough basic cell microscopy images and you fed the AI and AI now shows us the, what we were trying to do from this point back? And now there's this question, these are fine questions, and this is going to be the next step. So do you think that there will be a time that we won't actually need to use those microscopes anymore to do those imaging? So then you will actually need something even fancier, and we will stuck again because that microscope doesn't exist yet.
Right. I mean, obviously, you would throw more multimodal data as the questions get beyond the scope of the data that we have. There might be other microscopes that come along, but I think the number of things that we don't know, that we could know, with the tools we have now is vast. And so the day, I'm not saying that we shouldn't make more tools. That's what I do for a living, okay? But I do think that the thing that has held us back is the fact that we can't make sense of the data we can collect today, okay? And so that's the first task.
Hi, very impressive talking. So I'm focused on the fluorescence lifetime imaging. So I'm a little curious about, is it possible or hard to combine fluorescent lifetime imaging with light sheet?
No, it's possible. I mean, it's more hardware in order to do it, but it's potentially doable. Yes.
Yeah, because I think if we combine together, it could reveal more information than just focus on the intensity, right?
Yeah. There's certainly something in it too. Yes.
Okay. Thank you.
Mm-hmm.
Thank you for the amazing talk. I guess I want to maybe come back to the previously raised question. So what do you think we, as let's say the microscopy community, could do to actually make the data that we generate more actionable? And, kind of to address the gap that you also mentioned, right? That we seem to have the tooling to do a lot of amazing things with this data. We seem to have a lot of amazing data, but somehow it just doesn't happen, right?
Yeah. Again, my answer is to work with parties who are competent at AI to be able to make interpretable versions of what comes out of our microscope. Like I said, our brains work in 2D or 2D plus time, okay? The models can work in 4 and 5D. And so I think this is an obvious choice of marrying two technologies because it's the right technology to get past the problems we have of this interpretation.
Thanks.
Over here. I guess, so you presented the final observatory vision that the LLM would be kind of used for querying the data set. Or to what extent do you think you could just kind of let the LLM loose on it and go through chains of reasoning or hypothesis generation and just fully explore it? Because as you said, we might be limited by what we can think of, but it might be.
You can just look at one of those movies and just you with your little ML up here can generate 100 hypotheses from a tiny fraction of one of those movies, right? And so, if the architectures become sophisticated enough to do that work for us, great, it'd be wonderful.
So Eric, we had this discussion yesterday in the discussion panel of, if you had to choose between time or an extra modality, what would you choose?
Yeah, I told you my choice right here.
Exactly. Well, it's also interesting that many people chose time.
Yes.
As a critical parameter in biology that's information rich. But so I know your choice. Mm-hmm. I guess in that graph of data gathering, that was 4D information.
Yes.
So that is with one color.
One color.
That's not the fifth color.
That's right.
So I guess my question is, like, if you had to choose like a fifth color or some like clever way of, you know, getting more biology into that data set, rather than cell outline, for example.
Right.
Do you have any like clever ideas of how to?
Not exactly. I think again, color number 5, right, is where a lot of the biological questions lie after you have the segmentation, right?
Yeah.
And I don't see an obvious way of doing anything except, you know, doing some additional pre-training and fine-tuning for the specific task for specific organelle interactions or you know, or lifetime or whatever, right? Yeah, go ahead.
Well, just to follow up on that, you know, because time is also, you can sample at different rates.
Yeah, and they're all important, right?
They're all potentially important.
Yes.
But maybe not. You know, maybe, you know,
depends on the question.
Yeah.
I mean, I don't need 10 milliseconds if I'm looking at development, but I need 10 milliseconds if I'm looking at single molecule kinetics, right? So fortunately, the imaging platform itself has a broad enough dynamic range to give data for both of those questions. But the models have to then, at some level, I'm not sure whether the models would, this is just total speculation, the guy doesn't know anything, but whether the interval, the number of the interval between the model matters so much as the ratio of the sampling interval to the speed of the dynamics that's happening, right? Is that at a certain level, single molecules moving around at 10 milliseconds isn't a different problem to the model than mitochondria moving around at tens of seconds, okay? Or at least I'm bullshitting that point right now. I don't know to what extent that will make our life easier or harder in terms of training.
Eric, I love the vision. I think my question follows up what Ron was asking you also, which is that you're going to have a limited numbers of channels you can follow in time. How are you thinking about prioritizing what to image, right? You're focusing a lot on morphology. I mean, if I want to understand how the brain works, looking at neuronal morphology is not going to get me very far, right? So you see what I'm asking? When you do the math of like how scalable that is, how are you thinking of strategizing what we should image to answer the biological questions?
Let me take a step back, okay, so when I try to put this vision together, one of the things I think about is what if we fail and where do we fail, right? And is there a booby prize of sorts, right? Okay. So the first thing that we get is A, we will get a library of transgenic organisms. B, we'll get a crap load of data, which people can, be all open source, right? Beyond that, if we can just get past the segmentation task, then what we've done is we've taken cell biology away from the cover slip. Because at that point, anybody with whatever reagents they have, if they can do microinjections into the fish, that's easy, right? Then they can take their fish with our membrane label, they can computationally with the models separate all those cells. They can understand every cell themselves just as if they were looking at cells on a cover slip and do the exact same type of biology they do today, but with the confidence that they're doing it in a more physiologically relevant context and with the metadata that tells them where it is and how that might change. I think that alone would be revolutionary. And I think that's the low-hanging fruit to try to do this. And then beyond that, then it gets much more into science fiction from there, right?
Okay, one last quick question. Go ahead. Yeah, you were there.
Oh.
Yeah, go ahead.
Do you see a path? This is amazing if we work on the organisms or cells, but do you see a path that the fluorescent microscopy can be used for human, or in vivo, or what are your suggestions? Like maybe some like label-free.
In vivo potentially in large organisms. One could eventually, you look at what's happened with 2-photon and freely moving animals with 1 gram things on their heads and like that. There's the potential to think of endoscopes that would use this type of technology to be able to do things in organisms at some point. I think plausible, yeah.
All right, thank you very much, Eric.